Welcome to Satu Carry Online Nursing channel. In this video, we are going to discuss the Physiology question paper of 2018 July exam held conducted by Pandit B.D. Sharma University of Health Science, Rohtak for BSc Nursing first year students. So the Physiology questions were divided into three parts. In this video, we are going to see the first part of Physiology question papers, which contains three questions. The first question about spermatogenesis, the second one factors regulating cardiac output, and third one is Juxtaglomerular apparatus. These are the three questions the student has to attend compulsorily. There is no choice for these questions. Now we see how you need to present in your final exam. The first question is spermatogenesis. How we want to present in the exam is very important. So this lecture video will guide you the way you want to present in your final exam. Okay, we go to the first question, spermatogenesis. You know, the spermatogenesis is a process by which the male gametes called spermatozoa, sperms, are formed from the primitive spermatogenic cells, which is called as spermatogonia in the testis. Spermatogenesis takes place in the testis. In this process, the male gametes, known as spermatozoa or sperm, is developed from the primitive cells called as spermatogenic cells or spermatogonia. It takes 74 days for the formation of a sperm from the primitive germ cell. So 74 days the process is going on for the complete development of sperm. Spermatogenesis. In this, along with the sperm cell, one more cell is that is called Sertoli cell. This Sertoli cell is a cytoplasmic attachment of the spermatogenic cell. These cells supplies other materials for the development. The Sertoli cells supplies the needed material for the development of sperm cells. So the sperm cell and Sertoli cells are very important in the spermatogenesis process. What are the stages of spermatogenesis? There are different stages. First stage is stage of proliferation. Second stage, stage of growth. Stage of maturation is the third stage. And final stage is stage of transformation. These are the four stages in the spermatogenesis process. Stage of proliferation, stage of growth, stage of maturation, and stage of transformation. Now we see one by one in detail. The first stage is stage of proliferation. So before writing the detailed answer about spermatogenesis, this diagram you can draw. Spermatogonia, it is a primitive cell from which primary spermatocyte is produced, and this primary spermatocyte gives rise to secondary spermatocyte, and that develop into spermatid, and finally a spermatozoa is produced from spermatoids. This is the diagram you need to draw in the exam. Now we go to the first stage, stage of proliferation. Each spermatogonium contains diploid number of chromosomes. That is 23 pairs of chromosomes are present in the spermatogonium cells. The 23 pair includes 22 pair of autosomal chromosomes and 1 pair of sex chromosome. One sex chromosome which includes one X chromosome and one Y chromosome. During the stage of spermatogonia, what is happening, that is thus each in the stage of proliferation, the spermatogonium cells divide by mitosis, but there is no change in the chromosomal number. But the division occurs through mitosis process, but there is no change in the chromosomal number. The 23 pair remains intact. During this stage, the spermatogonia migrate along with Sertoli cells towards the lumen of the seminiferous tubule. So when you know when you study the anatomy of testis, you know the male reproductive system parts, various parts. You know the seminiferous tubule. So the proliferation in this stage, the spermatogonia cells along with Sertoli cells move towards the lumen is the opening of the seminiferous tubules. Next stage, growth. In this stage, the primary spermatocyte grows into a larger cell. Apart from growth, there is no other change took place in this spermatocyte. So just the cells are getting bigger in size. The third stage is maturation. After reaching the full size, each primary spermatocyte quickly undergoes meiotic or maturation division, which occurs in two phases. So in the third stage, once the cells get bigger in size, it undergoes meiotic division, otherwise called as maturation division. This occurs in two phases. In the first phase, each primary spermatocyte divides into two secondary spermatocytes. So this we have seen in the diagram earlier. At this phase, each secondary spermatocyte receives only the haploid or half of the number of chromosomes. So in the 23 pair, one set is going to each spermatocyte, that is haploid. What is happening in the second stage? In the second stage, each secondary spermatocyte undergoes second meiotic division. So the second meiotic division followed by the first meiotic division. It results in the similar cells called spermatids. So here the spermatids are produced. This spermatids also contains the haploid number of chromosomes. So in the first stage, you remember the division occur, but there is no change in the chromosomes. It remains same. But in this, it gets haploid. It becomes a half, divide into one. Another half goes to the another spermatid. The stage of transformation. The next stage. There is no further division. Spermatids are transformed into a mature spermatozoa by means of spermatogenesis and released by spermination. So in the transformation phase, there is no further division. The spermatids are completely transferred into mature spermatozoa known as sperms, and these sperms are released outside by the process called spermination. The development of sperm is called as spermogenesis. Spermogenesis is the process by which the spermatids become mature spermatozoa, and spermination is the process by which the mature sperms are released from the Sertoli cells into the lumina seminiferous tubule. So this is the answer what you want to give in the final exam if this question comes in the question paper. Okay, so hope you understood about spermatogenesis. Next we go to the second question, juxtaglomerular apparatus. So for many students this looks a very difficult question, but it is a easy question. Juxtaglomerular apparatus. So in order to study this question, you should visualize the picture of nephron in your mind. Juxtaglomerular apparatus is a specialized organ situated near the glomerulus of nephron. So juxta means near. So these cells are situated near the glomerulus of the nephron. Juxta meaning near. So imagine that structure of a nephron. So this part is called as juxtaglomerular apparatus. So this is you might have studied Bowman's capsule, and these are the part which is present near to the uh, where this is the juxtaglomerular apparatus which is present near to the uh, nephron. Okay, that is a specialized organ. So we have different structures like macula densa, and here we are having afferent arteriole and afferent arteriole, and we have various there is cells called as mesangial cells. So you will see one by one in detail. Now we move on to the structure of juxtaglomerular apparatus. It is formed by three different structures: macula densa, extraglomerular mesangial cells, and juxtaglomerular cells. So these three structures together form a juxtaglomerular apparatus: macula densa, extraglomerular mesangial cells, juxtaglomerular cells. Macula densa. It is the end portion of the thick ascending segment before it opens into the distal convoluted tubule. It is situated between afferent and efferent arterioles of the same nephron. You can see the diagram again. The macula densa is located between the afferent arteriole and efferent arteriole, and this is present in the where it is present. This present the end portion of the ascending segment of the nephron. Macula densa is formed by tightly packed of cuboidal epithelial cell. The cells found in macula densa is cuboidal epithelial cells. What is extraglomerular mesangial cells? They are located in the triangular region bound by afferent arteriole, efferent arteriole, and macula densa. So the extra means outside. So extraglomerular mesangial cell, which is located in the triangular region. So it is covered, bounded by three organs like efferent arteriole, and macula densa. These cells are also called as agranular cells because there is no granules, and these cells also another name is lattice cells or Gurmagitic cells. Gurmagitic cells. So these are the other names of extraglomerular mesangial cells. Next one is glomerular mesangial cells. Besides extraglomerular mesangial cells, there is another type of mesangial cell situated between glomerular capillaries called glomerular mesangial cells. It is also known as intraglomerular mesangial cells, which is situated between the capillaries, glomerular capillaries. So it is known as intraglomerular mesangial cells. What is the function? This cells supports the glomerular capillaries loops by surrounding the capillaries in the form of a cellular network. It provides a cellular network-like structure, and it supports the glomerular capillaries. These cells plays an important network in regulating the glomerular filtration by their contractile property. So the important function of the glomerular mesangial cell, it controls the glomerular filtration rate, and also it gives support to the glomerular capillaries. What are the another thing is glomerular mesangial cells also has the property of phagocytic function. These cells also secrete glomerular interstitial matrix, prostaglandin, and cytokines. This helps to prevent any infection entering inside. So these are the another importance of glomerular mesangial cells. Now, juxtaglomerular cells. These cells are specialized. These cells are specialized smooth cells situated in the wall of afferent arteriole just before it enters the Bowman's capsule. So these cells are present in the wall of afferent arteriole just before it enters the Bowman's capsule. These smooth cells are most commonly present in the tunica media and tunica adventitia on the walls of the afferent arteriole. You know the afferent arteriole which enters the uh, Bowman's capsule, the blood vessel, and the inner layer, the middle layer, tunica media, and outer layer, tunica adventitia. So these cells are present in this area. These cells are also called as granular cells because of the presence of secretory granules in their cytoplasm. So this is about juxtaglomerular cells. Now, to conclude the juxtaglomerular apparatus, the functions, it secretes two hormones, renin and prostaglandin. These two hormones regulates our blood pressure. Renin along with angiotensin controls the blood pressure. So this is how you need to answer the juxtaglomerular apparatus in your exam. Now move on to the third question. The third question is about factors regulating cardiac output. There are four factors which regulates cardiac output. First, you must know what is cardiac output. Cardiac output is the amount of blood which goes out of the heart during each contraction. So the four factors includes venous return, force of contraction, heart rate, and peripheral resistance. Let's see one by one. First one is venous return. Venous return is the amount of blood which is returned to the heart from different parts of the body. When it increases, the ventricular filling and cardiac outputs are increased. So by the name, return of venous blood. That the venous return means the amount of blood comes to the heart from inferior vena cava and superior vena cava is known as venous return. If the ventricular filling is more, that means the venous return is good and the cardiac output is also going to be good. The cardiac output is directly proportional to the venous return, that already I told you. When the venous return is going to be high, the cardiac output also goes to high. What is venous return? The venous return is depend upon five factors: respiratory pump, muscle pump, gravity, venous pressure, and sympathetic tone. These are the five factors which influence the venous return. First one is respiratory pump. It is the respiratory activity that helps the return of blood to heart during inspiration. So at the time of inspiration, there are some mechanism which helps to bring more amount of blood to the heart by the respiratory organs. So how it is also known as abdominothoracic pump. The respiratory pump is also known as abdominothoracic pump. Why? Because when we take air inside the lungs, the diaphragm goes down and it creates pressure in the abdomen. As a result of that pressure, increased abdominal pressure, it gives pressure to the veins, which takes the blood from the lower part of the body. As a result of that, the venous return is increased. During inspiration, what is happening? The thoracic cavity expands and makes the intrathoracic pressure negative. So when we take oxygen from the atmosphere during by the process known as inspiration, the thoracic pressure is going to be negative. At that time, the diameter of the vena cava increases, which result in the increased venous return. When the diameter of the vena cava is going to increase, more amount of blood is going to pass through the vein to the heart. So this is about respiratory pump or abdominothoracic pump. Muscle pump. It is the muscular activity that helps in the return of blood to the heart. During muscular activities, the veins are composed of compressed or skews. So when you do any physical exercise at gym, then automatically it gives contraction to the blood vessels and the muscles. Due to the presence of valves in veins, what will happen at the time of compression? Blood is moved towards the heart. So the valves helps the the wall helps to prevent the backward flow of the blood. So because of the compression of the vein, the blood moves in the forward direction, which enhance the venous filling. Venous return is more when muscular activity increases. When you do more workout, your muscle pump is going to be increased, which in turn increase the ventricular filling. As a result of the cardiac output also increases. The next one, the next one is Gravity. Gravitational force reduces the venous return. For example, if you stand for a long period of time, the blood which needs to come from your lower part of the body will not come very easily. The blood cannot travel easily against the gravity. That time the pooling of blood occurs. That is called as venous pooling. Venous pooling. Because of the amount of blood returning to the heart, because of this, the amount of blood returning to the heart also decreases. This is about gravity. The third one is venous pressure. Venous pressure also affects the venous return. You know that venous pressure in the small venules, the very minute portion of the veins called venules, is high, 12 to 18 millimeter of mercury. But when it comes to the smaller artery and the larger artery, and when it comes near the vena cava joins the right atrium, the pressure in the vein is decreasing. This pressure gradient, this pressure difference at every part of the venous tree helps us driving force of the venous return. This is the reason why the pressure difference in the venous tree, that is from the venules to the small artery, small veins and the large veins, what will happen? The pressure is going to decrease further. That leads to the increased venous return with a great force. Next one is sympathetic tone. Venous return is aided by sympathetic or vasomotor tone, which causes constriction of venules. So when the sympathetic tone increases, automatically the venous return also increase. Okay, so sympathetic tone is directly proportional to the venous return. The next one, the second factor, first factor we have seen that venous return. The second factor is force of contraction. Cardiac output is directly proportional to the force of contraction, provided the other three factors remain constant. So when the force of contraction of the heart muscle is going to be more, the cardiac output also going to be high. So in this, the other three factors should remain constant. The next thing is heart rate. Heart rate is directly proportional to cardiac output. Moderate change in the heart rate can either increase or decrease the heartbeat. Any mild change does not matter, but when there is moderate changes in the your heart rate, that will affect the cardiac output seriously, either in the positive way or in negative way. The last factor is about peripheral resistance. Very important to note peripheral resistance. Peripheral resistance is the resistance offered to the blood flow at peripheral blood vessels. So when the blood is reaching the peripheral blood vessels, it gives some resistance for the backward flow of blood to the heart. So it is the resistance or load against which the heart has to pump the blood. That is, against this resistance the heart needs to pump the blood. Then only the blood can return back to the heart. So the cardiac output is inversely proportional to the peripheral resistance. So these are the various factors which regulates our cardiac output. So hope you can write all these three questions in the exam in future if it comes. Now it's time to questions or doubt. If you have any question or you come across any doubt in these three questions, you can comment in the box given below so that I could reply to you as early as possible. Don't and thanks for watching my video on my lecture, and I request you all to subscribe to Satu Carry Online Nursing channel for more education videos. Hope you enjoy. Take care. Bye.